Welcome back to the another part of the video which we were discussing about the structure of DNA. So in the last video we were discussing about what DNA is, what are the different types of nucleic acid, particularly what are the components of DNA and what it is made of. We have already discussed. If you have not watched that video, I prefer you to first watch that video, first part, and then you can easily understand the properties of the DNA. So in this part of the video, we will be mainly focusing upon the types of DNA and certain features regarding the DNA. So let's discuss one by one. First, we will be discussing about the forms of DNA. So mainly the DNA are mainly present in the three forms, that is A form, B form and Z form. Okay. So B form is a usual form of the DNA which is mainly found in the organism. And the Z form of DNA is known as the left-handed, whereas both the A form of the DNA and the B form of the DNA is the right-handed form. So what is left-handed, what is right-handed? Let's see. So in this part you can see this is one strand. This strand is facing towards the right hand side and this strand is facing towards the right hand side of its place. Similarly, B strand is also facing towards the right hand, so one strand towards right hand side, another strand also facing towards the right hand side. Whereas in case of Z form of DNA, you can see both the strands are facing towards the left side. That's why they are called left-handed. And also the Z form is known as the zigzag form of the DNA. Why it is zigzag? Because the base pairs are arranged in a zigzag manner. That's why the DNA, this form of DNA is called as a zigzag DNA. And in this form of the DNA, their turn, that is the repeating unit of the base pair, will be 2 base pair per turn. That is, after every 2 base pair, a repeating unit will be seen. Whereas in case of A form and B form, only 1 base pair per turn. And what is the base pair, mean base pair per turn? That is, after how many base pair there will be a turn in the DNA structure. In case of A form, that is 10.7, B form is 10.4 and in Z form 12. After every 12 base pairs, there will be a turn in the structure of the strand. And what is the angle? That is 20 degree in case of B form, A form, sorry. B form is -6 degree and Z form is 7 degree. So these are the 3 forms of the DNA. Apart from this, there are also 2 other forms of DNA, that is triplex DNA, which is known as the 3-stranded DNA. What is 3-stranded? That is, one strand will be polypurine rich, another strand will be polypurine, another one will be polypyrimidine. And the third strand, you can see, I am darkening it with this one. This third strand will be present between these 2 duplex by the hydrogen bonding. And what in which case does this triplex form is mainly present? When there is a low pH. Okay, when there is low pH condition, in that time triplex DNA will be formed. And what is G quadruplex form? That is mainly guanine rich. Guanine mainly forms the square shape like structure DNA known as the G quadruplex DNA. Okay, they are also joined by the hydrogen bond. All the forms of the DNA are mainly joined by the hydrogen bond, which we have already discussed in the last part. Now coming to the main feature about the DNA supercoiling. What is DNA supercoiling? As the DNA is a long strand structure, so inside the chromosome they need to be present in a condensed manner or in a closed compact manner. For that, supercoiling takes place. What is supercoiling? Supercoiling is coiling of the DNA upon itself. Usually the DNA remains in a relaxed state. Okay, usually if you see the normal structure of the DNA, they will be in a relaxed state. But when they are present in a chromosome, they will be in a coiled state, that is supercoiling state. It can occur in both the circular DNA, that is in case of bacterial DNA is a circular DNA, linear DNA, that is in a eukaryotes DNA, they are also supercoiling takes place because they are the huge structures, so they need to be compacted. That's why they are in the coiling structure. If you take out the DNA, and normally if you see, they are in the relaxed form. Relaxed form of DNA is mainly 10.4 base pair per turn. After, if the base pair per turn is more than, if more helical turns will be present or if more coiling takes place, then it is called as positively supercoiled. If less coiling takes place, that is below 10.4 base pair per turn, is known as negatively supercoiled. Okay, mostly negatively supercoiling is seen in case of the normal organism. And how can we represent this DNA supercoiling mathematically? How can we measure that? By linking number. What is linking number? What are its property? Let's see. First we will see certain features regarding the linking number. So linking number is mainly defined in the covalently closed circular DNA that have fixed molecule present in a covalently present. Okay, and how can we change the linking number? We can change the linking number by breaking the phosphodiester bond present in the DNA structure strands. We have already discussed about what is phosphodiester bond, what is the structure of DNA. We have already discussed in the first part of the video. Do watch it. Next, to change this linking number, there are two types of enzyme. Why do we need to change the linking number? Because we have already discussed, I have already told that linking number is to measure the coiling. So to relax the supercoiling, topoisomerase enzyme is needed. And what is the role of topoisomerase enzyme? We will be discussing when we will be reading about DNA replication. What is the role of topoisomerase enzyme? It is mainly used to relax the DNA. Okay. So this linking number is mainly changed by the enzyme, that is topoisomerase. Topoisomerase is of two types, that is topoisomerase 1 if there is single strand break, topoisomerase 2 if both the strands of phosphodiester bond needs to be broken down. Okay, and linking number is majorly defined by the number of twists, that is the turn of helical turn. If you see, this is known as the twist. Okay, this is known as the twist. This is one strand, this is another strand of the DNA. Okay, this is known as the twisting. Okay, and what is rith? That is supercoiling of the DNA. This is known as the rith. That is supercoiling of the DNA is known as the rith. The sum of this is known as the linking number. Okay, now you might have been clear that twisting number, twist number plus rith number is total. We can know what is the linking number or what is the actual number. Okay, usually in case of circular DNA or in the relaxed circular DNA, the rith is 0 because there will be no supercoiling. So rith is equal to 0. So the linking number is equal to rith number. We can measure by this formula. Okay, by this formula we can measure the linking number. Okay, next this is all about the supercoiling. This is the feature about the supercoiling. What is DNA supercoiling? What are the different forms of the DNA? By this part of the video, we have come to the end of the basic structure and what are the basic properties of the DNA. In the next part of the video, we will be discussing about a significant performance which the DNA does, that is the DNA replication. There is a more beautiful process which will be discussing what are the, this may be three part. The DNA replication may be of three parts, that is what are the enzymes involved in it. In the first part we will discuss about what are the enzymes involved and what are the role of that enzymes in the replication process. Second part, what is the replication in prokaryote and replication in eukaryote. So in this we will be discussing each one in detail. So this is all for the today, and if you have not watched the first part of the video, do watch it, like, share among your friends as much as possible. I have made many videos as well as playlist. Do watch it, check it regarding vertebrate, invertebrate, evolutionary biology as well as physiology. I've made videos, and do share among your friends because these are very informative videos and very basic and mostly asked in the exam. So you, for the clear basic understanding, this video should be shared among your friends and comment if there is any doubt regarding this video. Thank you and have a nice day.